The overall goal of this procedure is to create a consistent and reproducible human brain tumor model that can be used for the accurate testing and comparison of novel therapeutic strategies. This is accomplished by preparing the animal for surgery and locating the correct site for injection. Once located, a hole is drilled in the skull and a syringe is lowered at the proper depth to obtain tumor growth. Cells are then injected in a slow, controlled manner, followed by a delayed and gradual withdrawal of the needle. Ultimately, a consistent mouse model with tumor growth histopathologically similar to the human condition can be developed, and growth rates can be measured by luciferase-based in vivo imaging.
The precision of this technique is vital to understanding new therapeutic strategies for human brain tumors. Variable localization of tumor injection can lead to changes in tumor growth that are due to microenvironment, drug delivery, or other things that vary greatly amongst different areas of the brain.
Visual demonstration of this method is important because the steps involving mouse positioning and injection site localization require three-dimensional manipulations which are not easily described in written or graphical format.
To begin, clean and decontaminate a mouse stereotaxic frame and all other equipment needed for surgery. Using settings optimized for the model chosen, program the injection parameters and other variables for the operation of the syringe pump. Next, arrange autoclaved surgical tools in the work area. Include at least two 1.0